Study carbohydrate metabolism, which belongs to glycolysis, gluconeogenesis, and pentose phosphate pathway. In lecture 6, course is BBT315 metabolism. So, metabolism means breakdown and synthesis. In some reactions, the macromolecule will be breakdown to a smaller one, is called catabolism, and in anabolic reactions, um, simpler molecule will convert it to macromolecules. Okay, so here the glycolysis means breakdown of glucose, and gluconeogenesis means synthesis of glucose. So this is catabolism, this is anabolism. Together with that, carbohydrate metabolism. Okay, so before starting, let's study glucose, which is a versatile molecule, and what are the fates of glucose. So, glucose occupies a central position in the metabolism of plants, animals, and many microorganisms. There are four major pathways. So, first, the glycolysis. So, glucose will be oxidized to a 3-carbon containing pyruvate to provide ATP and metabolic intermediates, and it is a short-term energy needs. And secondly, when there is an excess energy, glucose can be stored in polymeric form as a polysaccharide like glycogen and starch or sucrose. And thirdly, glucose can be oxidized via pentose phosphate pathway, 12-ribose 5-phosphate for nucleic acid synthesis, and NADPH for reactive biosynthetic process. And also, it is important for detoxification of lipids. Fourthly, the carbohydrate will be served as a structural polysaccharide like extracellular matrix, cell wall of bacteria, fungus, and plant. These are the four major pathways of glucose after ingestion in the diet. So, glucose is very important, and it is relatively very rich in potential energy, and thus a good fuel. And the complete oxidation of glucose to carbon dioxide and water proceed with a standard free energy change of 2840 kilojoule per mole. By storing glucose as a high molecular weight polymer such as starch or glycogen, a cell can store large quantities of hexose unit while maintaining a relatively low cytosolic osmolarity. Glycogen, being insoluble in water, will not offset the osmotic pressure, where glucose, being soluble in water, will disturb the osmotic pressure, which will lyse the cell. And when energy demand increases, glucose can be released from this intracellular storage polymer like glycogen or starch and used to produce ATP either aerobically or anaerobically. Glucose is also a remarkably versatile precursor capable of supplying huge array of metabolic intermediates for biosynthetic reactions. Organisms that do not have access to glucose from other source must make it. Photosynthetic organisms makes glucose by reducing atmospheric carbon dioxide to triose like glycerol 3-phosphate, then converting the triose to glucose. And non-photosynthetic cells make glucose from simpler 3 or 4 carbon precursor by the process of gluconeogenesis and effectively reversing glycolysis in a pathway that use many of the glycolytic enzymes. So let's study glycolysis. So in glycolysis, a molecule of glucose is degraded in a series of enzyme catalyzed reaction to yield 2 molecules of 3 carbon containing compound pyruvate, and some of the free energy released from glucose is conserved in the form of ATP and NADH. So glycolysis is an universal central pathway of glucose metabolism. The pathway with the largest flux of carbon in most cells. The glycolytic breakdown of glucose is the sole source of metabolic energy in some mammalian tissues and cell types like erythrocyte, renal medulla, brain, sperm, for example. Some plants tissues that are modified to store starch, and some aquatic plants derive most of their energy from glycolysis. Many anaerobic microorganisms are entirely dependent on glycolysis. Anaerobic degradation of glucose or other organic nutrient to obtain energy conserved as ATP is termed as fermentation, because living organisms first arose in an atmosphere without oxygen. An anaerobic breakdown of glucose is probably the most ancient biological mechanism for obtaining energy from organic fuel molecules. Glucose has breakdown of glucose or glycolysis has two phases. One is preparatory phase, another is payoff phase. The breakdown of 6 carbon containing glucose into 2 molecules of 3 carbon containing pyruvate occurs in 10 steps, the first 5 of which constitute a preparatory phase. And in the sequential reaction of glycolysis, 3 types of chemical transformations are particularly noteworthy. The first one is degradation of carbon skeleton of glucose to pyruvate. Second one is phosphorylation of ADP to ATP by compounds with high phosphoryl group transfer potential formed during glycolysis. And the third is transfer of hydride ion to NAD, forming NAD+. So let's study the preparatory phase, where the first 5 steps of glucose involved. So glucose, this is the structure of glucose. So glucose will first be converted to glucose 6-phosphate by hexokinase enzyme, where phosphate group will be supplied by ATP, forming ADP. This is the irreversible reactions. And glucose 6-phosphate then converted to glucose fructose 6-phosphate by phosphofructoisomerase. And then fructose 6-phosphate will be converted to fructose 1,6-bisphosphate by phosphofructokinase 1. And here ATP will be converted to ADP. That means the phosphate group came from ATP. And here you can see hexokinase. Glucose is hexose. So kinase is an enzyme that involves the phosphorylation reaction. So hexokinase means the enzyme that phosphorylates hexose. And here glucose and fructose are isomer. So the enzyme name phosphohexosisomerase. And the third enzyme is phosphofructokinase. Basically, this is a fructose and containing phosphate. So this is phosphofructose, and the kinase is the enzyme that phosphorylates the fructose, producing fructose 1,6-bisphosphate. And fructose 1,6-bisphosphate then finally converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by aldolase enzyme. And here you can see the fructose can be converted to 3 carbon containing 2 molecules. And then dihydroxyacetone phosphate can be converted to glyceraldehyde 3-phosphate by triose phosphate isomerase. So this is the first 5 reactions of preparatory phase, and where phosphorylation of glucose and its conversion to glyceraldehyde 3-phosphate will be occurred. And let's start the payoff phase. So the energy comes from payoff phase, and glyceraldehyde, from 1 molecule of glucose we got 2 molecules of glyceraldehyde, which then converted to 2 molecules of 1,3-bisphosphoglycerate by the reaction, by the name of the enzyme, or catalyzed by glyceraldehyde 3-phosphate dehydrogenase. It's generated from the name of substrate. So where phosphate will be induced in glycerol 3-phosphate producing diphosphate molecule, where NAD will be converted to NADPH. That means NADH will be reduced to NADH. The production of NADH is very, very important, because this NADH will then enter into the mitochondria. So glycolysis occurs in cytoplasm, and NADH produced in glycolytic reactions of glycolytic reaction in cytoplasm will enter into the mitochondria and then finally enter into the electron transport chain ETC, where it oxidizes to produce NAD, and then this NAD will come back here again and to facilitate the glycolytic reactions. That means facilitate the formation of glyceraldehyde 3-phosphate to 1,3-bisphosphate glycerate. This is for aerobic case in the presence of oxygen. But while in anaerobic respiration, that means when there is no oxygen, what will be happen? There is no oxygen, that means no electron transport chain there. So NADH will not be oxidized to NAD in that case. So what will happen there? If NADH will not be able to convert it to NAD plus in ETC, then the concentration of NADH will be higher and there will be no NAD here. So if there is a lack of NAD, this reaction will be stopped, because when glyceraldehyde 3-phosphate will donate this proton, there will be nothing to accept the proton here. So the reaction will be stopped. But to continue the glycolysis, this NADH2B is very important to be reduced, to be oxidized through other reactions, like in anaerobic respiration, when pyruvate is converted to lactate. In that case, NADH will be used to produce NAD+. Then this NAD plus will come here again to continue the glycolytic reactions here. And then 1,3-bisphosphate will be converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase, where 2-ADP will be consumed to produce ATP. This is the first substrate level phosphorylation to produce ATP. Then 3-phosphoglycerate will just change the phosphate from number 3 to number 2 position and will form 2-phosphoglycerate. And then 2-phosphoglycerate will be converted to phosphoenol pyruvate by enolase enzyme. And phosphoenol pyruvate then finally converted to pyruvate by pyruvate kinase. This is the second step or second ATP forming reactions or second substrate level phosphorylation, where 2 ATPs will be formed. This reaction has been shown here in linear form. Okay, so what is the summary of the glycolysis? The summary of the glycolysis is glucose, NAD, 2 molecule of ADP, 2 molecule of inorganic phosphate will produce 2 molecule of pyruvate, 2 molecule of NADH, 2 molecule of ATP, and water. So used molecule is 1 molecule of glucose, 2 ATP and 2 NAD, and made 2 pyruvate, and that has various different fates, and 4 ATPs used for energy requiring process within the cell, and 2 NADH must be reoxidized to NAD in order to continue glycolysis, that I said already. So glycolysis is heavily regulated to ensure the proper use of nutrient and to ensure production of ATP only when needed. So the net production of ATPs in glycolysis is 2 molecules of ATP. Okay. So here we studied the glycolysis from glucose to pyruvate, and this is the first step of carbohydrate metabolism that occurs in the cytoplasm. Okay, that's all for this lecture.